Hi friends, today I will be described about the shotgun sequencing methods. Okay, what is their applications, what is their benefits, and what is the approaches to shotgun sequencing methods. Okay, so this cannot be escaped from the Sanger DNA sequencing methods. Shotgun sequencing method is used to sequence larger genome size, even human genomes. You know that shotgun sequencing method first sequence complete genome of the Haemophilus influenzae. Okay, so by the larger genome size we cannot sequence by the Sanger methods or Maxam-Gilbert methods. So require shotgun sequencing method. What is their approach and how Sanger DNA sequencing method is required in shotgun sequencing methods? Okay, so suppose that what is the mean of shotgun? You know that there is a gun, and when you shot anything by the guns, then different fragments will be obtained of anything which is shot by the guns. Suppose that there is a gun. Okay, there is a gun. When you shot anything in front of them, they can be fragments in continuous shot by the gun. It will get many fragments. That's how anything in front of gun. So there is also that may be used sonications or anything in the form of gun that will shot the larger genomic size DNA sequences. Larger, okay. That will genome sequence that will gun by the shearing with the help of sonications, ultrasonic waves that will get many fragments, and suppose that it will get larger fragments that will be 100 kb or it may be below 100 kb. Okay, that may be below 100 kb. Then this 100 kb sequences is cloned. Where clones? Bacterial artificial chromosomes. Because bacterial artificial chromosomes can carry 100 to 200 kb of the DNA fragments. And so, if you do not know about the cloning, then please go back to the cloning. That's how cloning is performed. Then come back to the shotgun DNA sequencing methods, because bacterial artificial chromosomes carry 100 to 200 kb of the chromosomes. Okay, so this is 100 kb fragments. Okay, all these fragments will be cloned by the bacterial artificial chromosomes back. Okay, how it will be inserted and how it will be ligated, it will another part of the topics. So today you will know that this bacterial artificial chromosomes will duplicate or clone the 100 kb. So all these fragments will be cloned. Okay, clone. I am showing only some drawings. Okay, this is 100 kb. So when the bacterial artificial chromosomes is introduced with this fragment sets, the bacteria will multiply, then the plasmids, bacterial artificial chromosomes will be multiplied. So the number of bacteria will increases, the number of these fragments will increases. So large number of these fragments will be obtained. Once out, large number of, there is many bacteria will divide, and with dividing these chromosomes will also divide, and the fragments will also clone. So the large number of chromosomes will be fragments will be obtained, which is shared by the shotgun method. Okay, so this is 100. Point out, this will aligned. The points term is alignment. Canting formations. What is the canting formation? Suppose that there is many fragments, so there is not single DNA sequences, more number of genomic sequences is available. So number of, suppose that there is 1, and within this there is another 2, and there is another 3 large DNA of the same size. Any human genomes in a cell, suppose that's you take me, examples. One cell contains a genome and many cells contains same genome sequences. So this will share obtains randomly sequences. Okay, randomly sequences. So it will be that, suppose that's A T G C T A. That is, you will take simple, for examples I am taking a fragments. You will mean that this is the genome. You will only mean for genome, not this is genomes, but for understanding T, C and A. Okay, it will, this is genomes, it will fragments different many types of A, C, T, A, T, G, C, T and A, and so many, that type of same sequences. Okay, so this will share, some fragments will obtain here, some will fragments will obtain here, and some another is A, C, T, A, T, G, C, T, A. That is same random fragments, because sort gun by sort will not recognize specific sequences. So some fragments will obtain from here. Okay, some will from here and some will from here. Okay, so you will point out there is many fragments. The same, this is this and there is this and there is this. So if you get these sequences and if you get these sequences, there is fragments, there is fragments. Okay, so points out then TAT, that will match, that will match. Okay, and this will fragments will obtained from here. So there is match, and these fragments will align with these fragments, that is contig. So from here there is contig formations, and these sequences will also gains, and another the fragments will obtain here, suppose that's here. Okay, there is another fragments here, points out because this will match, and there is another fragments obtained. So you will points out in these sequences there is matching. Okay, that is TA, TA, TAT, TAT. So at the time of alignment, the sequence will align and contig formation will take place by this way. There is here. So this sequence will easily recognize this sequence, and there is also TA, TA, and so all these fragments of the sharing sequences will be aligned, will be formed as contig. So this is the, in case of larger 100 kb, this is 100 kb, and the alignments will obtain as like this. Okay, as like this, because that is matching, there is a longer, and this may be also longer. So by this way alignments will be obtained. But even it is larger, this 100 kb is larger, because you know that 800 base pairs is sequenced by the Sanger DNA sequencing method. So this is also large. What happens? This will again, these fragments will obtain 100 kb. Okay, 100 kb will again fragmented. Okay, again fragmented. So many fragments will obtain, many fragments of this. Okay, and this will be near about 1 kb fragments. So here as a way, that's 1 kb will be obtained of this 100 kb. Okay, this is 100 kb. Okay, so this will again aligned by this way. Point out, this will again aligned by this way and sequenced by the Sanger DNA sequencing method by diadeoxy methods. You know. If you do not know Sanger method of DNA sequencing, then go back to the JK biology and see the Sanger method of DNA sequencing. Then you will find easily. That's how we will be sequenced of any 100 kb or 1 kb or below 1 kb DNA sequences. Okay, so this is 1 kb with sequenced by the Sanger method and then aligned by this way. So after alignment of these sequences, I will get or you will get of that sequences 1 kb. Okay, 1 kb. So this again aligned, this will aligned here 1 kb and again this will align to get these sequences after alignments. So by the shotgun DNA sequencing methods, this is smaller and smaller, and these smaller alignments will take place to make a larger size, larger fragments, larger chunks of the DNA. Okay, smaller chunks then align to larger chunks, and then even the larger will align to form complete genome of any organism. Okay, complete genome of any organism. So by this way, shotgun sequencing methods we can align. That is similar to the clone by clone sequencing. Why is it called clone by clone sequencing? Because you know that there is sharing of the fragments is cloned in the bacterial artificial chromosomes, and this will again points out there is, I have left one thing, that's the smaller fragments also required to clone. So this will be cloned into the plasmid of E. coli. Okay, the plasmid of the E. coli. Why? Because require large number of copy of these fragments. Okay, number of copies of these fragments. So even this will be cloned into the E. coli, because E. coli plasmids carry near about 1 kb of the fragments to clone, but bacterial artificial chromosomes carry what? This is, but plasmids carry 10 kb of the size or below 10 kb. Two duplicates are to clone, but bacterial artificial chromosomes carry 100 to 200 kb of the fragment size. Okay, so this will again cloned, and after cloning we require to sequencing by the Sanger methods. After sequencing, this will be aligned, and 1 kb will be obtained, 100 kb will be obtained, this 100 kb, and this will again align to form complete genome sequences. So shotgun sequencing method is used to sequencing a larger genome size. Okay, so this is the big approach, and but there is something drawbacks about the shotgun sequencing method, that is highly expensive. This technique is highly expensive in comparison to the another sequencing methods. Okay, even clone by clone sequencing. Why is it called clone by clone sequencing? Because this fragments is cloned into the bacterial artificial chromosomes, and this will again shear and subclone into the E. coli. So there is one clone and there is also second clone. So called clone by clone sequencing methods. Okay, clone by clone sequencing methods. So this is the expensive techniques to sequence, and first sequence was Haemophilus influenzae, and then human genome was also sequenced by this way. So hope this lecture would be beneficial for you, to understanding for you. So if you like this lecture, then please click on subscribe button and also share for your friends. Thank you.